let's start with a new chapter in biology today, Watson-Crick Model of DNA. So let's subscribe this channel first. DNA Double Helix structure. We begin with the most iconic image in biology, the glowing double helix. DNA is not straight like a ladder lying flat. Instead, it is twisted into a spiral form. This spiral shape is called a helix. And because there are two strands twisted together is called a double helix. This structure explains how genetic information is stored and copied. To simplify, imagine a ladder. The two long vertical sides are the backbones. The horizontal steps are base pairs. Now twist that ladder gently, that is DNA. This analogy makes the structure easy to understand visually. The rungs of the ladder are not random. They follow strict pairing rules. This rule is known as complementary base pairing, meaning each base pairs with only one specific partner. Adenine A always pairs with thymine T, no exceptions. This is due to chemical compatibility and hydrogen bonding. Guanine G always pairs with cytosine C. Again, this pairing is fixed. So the universal rule becomes AT and GC. This rule is fundamental to genetics. A and T are connected by two hydrogen bonds. Hydrogen bonds are weak individually, but together they provide the stability. G and C are connected by three hydrogen bonds. Because GC has three bonds, it is slightly stronger than A and T. This difference affects DNA stability. DNA strands run in opposite directions. This arrangement is called antiparallel. It means one strand runs upward while the other runs downward. Each strand has chemical ends labeled 5' and 3'. One strand runs 5 to 3, the other runs 3 to 5. This directionality is critical for replication. The vertical sides of the ladder are made of alternating sugar and phosphate molecules. This forms the backbone of DNA. The backbone protects and supports the internal bases. Phosphate group link sugars together. They create strong covalent bonds that stabilize the structure. The sugar in DNA is called deoxyribose. It gives DNA its name, deoxyribonucleic acid. In 1953, scientists proposed this double helix structure. This became known as the Watson-Crick Model. It revolutionized biology. DNA is located inside the nucleus of eukaryotic cells. The nucleus protects genetic material. DNA is extremely long. To fit inside the nucleus, it wraps around proteins called histones. When tightly packed, DNA forms chromosomes. Chromosomes are visible during cell division. DNA consists of two strands, not one. These strands are complementary and interdependent. During replication, hydrogen bonds break. This allows the strands to separate. When two strands separate, they form a Y-shaped structure called a replication fork. An enzyme called helicase breaks hydrogen bonds and unzips DNA. Think of it like opening a zipper. Another enzyme, DNA polymerase, builds new strands by adding complementary bases. It follows base pairing rules strictly. Because of antiparallel structure, one strand is synthesized continuously, leading strand, and the other is synthesized in fragments, lagging strand. The lagging strand forms short pieces called Okazaki fragments. These fragments are later joined together. Replication is called semi-conservative because each new DNA molecule has one old strand, one newly formed strand. Scientist Chargaff discovered amount of A equals to T and amount of G equals to C. This supported base pairing theory. The twisting creates grooves, major groove, minor groove. Proteins bind here during gene expression. The sequence of bases A, T, G, C forms the genetic code. This code determines traits. DNA acts like a blueprint. It contains instructions to build and maintain an organism. Your eye color, height tendency, and many traits are influenced by DNA. If we zoom in, body, organ, tissue, cell, nucleus, DNA. DNA is double-stranded. RNA is single-stranded. RNA contains uracil instead of thymine. Thymine exists in DNA. Uracil replaces it in RNA. DNA is a macromolecule made of repeating units called nucleotides. Each nucleotide has sugar, phosphate, nitrogen base. The model was proposed in 1953. It became one of the greatest discoveries in science. Base pairing ensures accurate copying. Without this rule, life could not continue. Replication begins when strands separate. This is essential for copying. Multiple replication forks form a bubble-like structure. Students must practice neat labeling of backbone, base pairs, 5 and 3 ends. Always remember the twisted ladder analogy. It simplifies the complex structure. A gene is a specific segment of DNA. that codes for a protein. DNA, RNA, and protein. This is the central dogma of molecular biology. 4 bases: adenine, thymine, guanine, and cytosine. The backbone protects the bases inside. Antiparallel structure enables replication accuracy. DNA stores information chemically. The order of bases is the message. Hydrogen bonds are weak individually but strong collectively. They allow flexibility. Steps of replication: unzipping, base pairing, and strand formation. Even one base change can cause mutation. The double helix is elegant and efficient. Incorrect pairing causes errors. DNA polymerase proofreads. DNA is organized but dynamic. During division, DNA condenses into X-shaped chromosomes. Replication ensures continuity of life. The double helix replaced earlier incorrect models. Directionality controls enzyme function. 1953 changed biology forever. This theory explained structure, replication, and genetic inheritance. Rungs of the ladder: base pairs form the rungs. Backbone forms the sides. DNA is not just a molecule, it is the foundation of heredity, evolution, and the life itself. So the Watson-Crick model explains double helix structure, complementary base pairing, antiparallel strands, semi-conservative replication, genetic information storage. This model connects chemistry with life. From microscopic molecules to entire organisms, DNA carries the story of life. So for the more bio sessions, please subscribe this channel.